Okay, I am showing it is noon Eastern, so I will go ahead and get started. We have hit recording. Hello there, and welcome to everyone. We'll give folks just another heartbeat in a minute, and I'll get through the preamble and some administration, and then we'll get started. My name is Ruel Hayden, U.S. Marketing Communications Manager for Nufarm Turf and Ornamental. I'm very happy you could join us for this Friday discussion and presentation. This webinar reviews the biology of take-all patch and discusses both cultural and chemical management practices available to golf course superintendents and turf managers. Information about new chemical management tools will also be presented. Participants will receive 0.1 education points with GCSA for this webinar. Before we begin, of course, a bit of admin. They are on by default or off by default, but please keep your microphones in mute and your cameras off. You are welcome to put your questions in the chat as they come to you, and we plan on having time for Q&A at the end of the presentation. The section is being recorded and will be made available on demand. If you have additional questions or follow up on the information or technologies presented briefly, please do so with Rick or reach out to your Nufarm Regional Sales Manager. You can find their contact information and contact us by visiting nufarm.com/USTurf/contact. One of Rick's slides will speak to this. And now, without further ado, let me introduce our speaker. I'm joined by my colleague and our presenter, Rick Fletcher, Technical Development Manager for Nufarm. Rick is a 40-year veteran in the turf and ornamental markets. He joined Nufarm in January 2013 in the role of Technical Service Manager. He is part of an excellent team of folks responsible for all aspects of Nufarm's product research, development, and field support here in the U.S. Prior to Nufarm, Rick spent 20 years serving as Director of Product Development for the Cleary Chemical Corporation. He holds a Bachelor of Science degree in Plant Science and a Master's in Agronomy and Plant Physiology from Rutgers University. So please join me in welcoming Rick. Rick, welcome, and over to you.
Thank you, Ruel. All pleasure that you're here today, and we've got a lively presentation for you that we will go through to discuss take-all patch. As an agenda, if you would, we've done our introductions. I wanted to go through some learning objectives with you. We will talk about the disease symptomology, what you're seeing, what is there on the course. We will talk about the biology and the characteristics, and in key, what's the effect of those things on your management strategies, because you need to know the critter before you attack the critter. We will talk about the cultural and chemical management strategies which have been developed over time for this pathogen, which will give you some tools to try to counteract this problem or to suppress it over time. And lastly, there'll be some time for question and answers. And of course, if you would use the raise hand, and Ruel will be monitoring that. So we look for some learning objectives, and I really want to hope that by the time we're done that you gain an awareness of the visual symptomologies. I'm sure most of you are quite aware of this, but also thinking about a working understanding of the biological activity of this organism, and in particular its life cycle. And we'll get into that in detail because that is going to directly affect your management choices and what you choose to do with this. I also hope to give you an understanding of the symptomology and why it attacks, what it does, what are the outcomes of its feeding on your grasses, and what you can do against that. I hope to give you an awareness of the data that is out there from universities. As you'll find, if any of you have done any searches, take-all patch is an elusive critter. This organism is hit or miss. We know about its characteristics, which we'll explain on younger turf species or environments that have had some drastic change. But we do know that when it does flare up, if we're fast enough, we can get some research. And of course, we'll talk about that research and give you some guidelines on which chemistries, which location strategies for the chemistries are going to be best. And lastly, I want to time out or give you this whole timing aspect of when is this organism active? When does it attack grass? What are the best times to treat it? Are there times that we really are wasting our time treating it? And really tie that in so that the whole package, if you would, is tied together on achieving the best strategies for managing this disease. So a little fun with the audience. I have fun and show you some pictures here of common things. And of course, I bet most of you can identify what you're seeing here. If you look on the top left, you know that that's fairy ring. If you look at the top right, some of you may figure out that that's actually a picture of summer patch. And then, of course, if we look at the bottom center, we know that that's the circular type of symptomology that we would see for take-all patch in a bent grass stand. Now, I have fun and ask, what are the similarities of these three diseases? They're made with different organisms, or are they? So we know that fairy ring is a basidiomycete complex, a Rhizoctonia type of organism lives in the soil. We know that summer patch is a soil inhabiting, root infecting organism that is caused by an organism called Magnaportha. And then we know that take-all patch is caused by a separate soil microorganism in the Gaiamannomyces. And we'll talk about the relationship between the summer patch and the take-all in a little bit, because that'll be curious for you. But my point in showing you this is that all of these are soil inhabiting pathogens, which at one time are interfering with normal root function, creating, in essence, dysfunction in the root. And of course, once you create dysfunction in the root, you end up with events which tend to show themselves during stress periods, which can lead to plant death. So the infection occurred and then the symptoms occur. So, and that's one of the key things about these soil inhabiting pathogens that we have to remember, is that when we see the symptomology, we may be way late for actually doing effective treatment. So the nuts and bolts of this disease, we know that this is a crown and root disease. We know it particularly likes bent grass in our environment. We know once in a while it can affect bluegrasses. We know once in a while it can affect fine fescues. But for our world, it's particularly attacking bent grasses on greens, tees, and fairways. It doesn't care. The causal organism in the Gaiamannomyces is a natural soil inhabitant. It tends to live as a saprophyte in the soil. In other words, eating just enough not to cause damage, but to have this happy medium. But then under certain conditions, that disease can get more severe. The feeding activities can get more severe, and then we can get this root dysfunction leading to the visuals in the foliage that we see. When we look at where it lives, it can live just about anywhere in the US, because there are cousins of this pathogen which also attack the cereal grains that we might think of. There's one that attacks rice, one that attacks wheat, one that attacks oats, and they're all closely related. So the spread of this is across the US. But when you look at our world in the Gulf situation with bentgrass, you're particularly looking at the Northeast, the Midwest, the Mid-Atlantic, the Pac Northwest areas, where we get these high concentrations of bentgrass being grown, as well as we get the typical fall and spring conditions for when this organism can rapidly grow. And I mentioned earlier about the relationship with summer patch and take-all patch. And one of the key things that's cute is that the Gaiamannomyces species is in a genus which belongs to Magnaportha. So think about that, or keep that in mind when we discuss product choices and chemical location strategies during treatment, and how those two are going to interact, because you're going to find some very similar coincidences between these two organisms, and it's likely to do with their relationship. So when you see these symptoms, and you've all seen these symptoms, this could be early June, this could be the first sign of high temperatures and moisture stress, that all of a sudden you get this circular patch, this colonized area, which will start to collapse. The leaves, the foliar tissue on top will start to collapse down. And you start to look at it closer. You may get out your soil profiler, cup cutter, and you'll pull out some roots. And what you'll see is that you'll see this discoloration. And of course, that discoloration is the root dysfunction that we're talking about, the infection of this organism, the colonization on the outside as well as the inside of the roots, which is starting to affect the root function. And of course, as we see in fairy ring, we have an organism that is changing the biology, which then affects the root's ability to get water through ammonia and hydrophobic zones. As we see with summer patch or Pythium blights, we see a root dysfunction, which then shows up in the plant can't meet the water demand placed on it with the sudden temperature changes. So we have this situation where we get this root infection, and that root infection relates to the dysfunction, which then relates to the foliar collapses that we see at the first sign of stress. If we were to look closer at those roots, we would see this type of browning, this discoloration on the outside as well as the inside of the roots. And the way this guy starts from the soil, and it should be mentioned, and I'll talk about this again, that this is an historic disease. It's a natural occurring organism. It lives from year to year in the soil. So if you had it last year and conditions are right, you can get it again the following year. And the way it survives is by making these fungal masses on the outside of the roots. And of course, as those fungal masses, ectotic outside hyphal strands, form on the root, then eventually they will go ahead and penetrate into the root and start to feed. So you have fun and ask some questions as far as, why are these things here? Why are they suddenly attracted? We know about their saprophytic nature, and I'll get into that in a second. But we would group these into what we would call the ETRIs, or the ectotic root infecting pathogens. And as I mentioned before, some of the other fungal organisms that are in this same group would be things like summer patch, things like Rhizoctonia root rots, things like Pythium root rots. And if we're on the ornamental side, things like Palaeviopsis. These are all soil fungi that attack the roots, form a mass on the outside, start to penetrate in, start to feed, cause root decline, result in the damage that we see. If you looked at the life cycle, we know the life cycle is pretty straightforward. We really don't have the life cycle drawn out for bent grass. So this is a picture of what it looks like on wheat, but it's going to be very similar as far as the infection. This is an ascomycete, so not to be too nerdy for you, but ascomycetes would be things like anthracnose, things like dollar spot are going to be in that same group, which means they have the ability to produce spores. And we know in the case of basal anthracnose that it can produce spores. And we know in the case of dollar spot that we don't see spores. And in the case of take-all patch, we haven't seen any direct infection that we can talk about related to spores. So what does this mean? This means, as I talked about before with this disease being historic, that this is surviving as vegetative mycelium. So this is living on the roots of the plant and surviving year to year. And as new plants put out new roots, or if you think of the fall going to the spring, and we get the resurgence of growth in our turf plants where they're putting out new roots, they're using those stored carbohydrates from last year, this organism will then go ahead and start to attack those new roots and complete or continue its life cycle as a saprophyte. So we think of the conditions that we know about from numerous examples in the field, from the limited research that's been done on university studies. And what we can do is say one thing that's very clear. This organism is very much in tune with its bent grass host. And we'll talk about this a little later in the timing, but I want you to think for a second along these lines. We have the normal cycles of bent grass, which is dormant. It then gets soil temperature and day length cues, and it starts to grow in the spring. We know that bent grass will typically start growing, let's say, 50 degrees Fahrenheit, and will grow quite well up to about 85 or 86. Why does it stop growing well at 86? Well, it's a C3 photosynthetic plant, which means its photosynthetic abilities start to diminish as we get close to 86 and pretty much stop past 86 degrees. And I want you to tie that into that photosynthetic engine and relate to this, that what this guy is doing is relying upon the plant to be active. And remember that the whole point of this plant activity is to make photosynthetic sugar to grow the plant better. Well, part of that growth situation is that the root will leak certain sugars, and this organism, as well as other organisms, will attach to those sugars and form an association with the plant. So we're going to see that the timing of the activity of this organism of take-all patch is very much in tune to the host as far as when it's producing sugar and when it's not producing sugar, both in the spring and in the fall. So we look at common activity ranges, and we can see anywhere from 45 to 85 degrees, but we know from numerous studies that it's in its sweet spot around 18 Celsius or 64 degrees under standard moisture conditions. So when we think about the timing of treatments for these, most university pathologists have settled on this range between 55 and 65 degrees as a soil temperature treatment target. Now, remember earlier I said this was a cousin of summer patch. When do we want to treat for summer patch? In that same temperature regime in the spring. So you can see the relationship with these guys, but there is going to be a sweet spot that we want to consider temperature when this is active to prevent this disease. Remember, we're trying to prevent inoculation or feeding on roots so that the dysfunction doesn't occur and the plants can get enough moisture, and the visuals that we see of healthy plant, a healthy playing surface, can remain. So if we look at what this looks like in a lab, and I can show you lots of neat pictures of this, but we get these temperature activity periods, the fungus will go ahead and start to initiate its growth and colonize these roots. We know that these roots live on the outside and that the mycelium will quickly spread across the roots and then eventually penetrate into the stele of the roots. And of course, remember, the stele is that vascular bundle that controls the xylem, the phloem, the cortex, etc., which is necessary for root function. And in this picture here, they took bent grass and went ahead and put the take-all patch on an auger and then put that auger underneath. And as you're watching the roots grow down, you're watching the take-all patch grow side to side here and spread into these roots. And this is the same thing that's going on in your soil profile on your green. So then once that goes ahead and colonizes the roots, of course, it proceeds inside the root. This is a great picture from Steve Thomas showing in these little dots here, these little blue arrows, where you can actually see the streaking of this organism inside the root, causing dysfunction as it's feeding. So again, this is all about trying to prevent this disease from doing this, because once we have root dysfunction, the only way that our plant can recover is by growing a new root. And if it grows a new root into an environment where this soil inhabiting fungi is living, then what's going to happen is that new root is also going to be infected. So we have to be very conscious that I can't drive that point home enough as far as this is a prevention idea. This is not a curative idea at best. So we talked about those temperature regimes, and of course, the timing of this talk was we hoped to have it a week earlier, but we couldn't pull it all together. But the timing is very critical, and I'll give you several examples here of what's going on right now. So this was a snapshot last year from Rutgers University. I had asked Ming-Yi if he would give me the measurements of his soil temps there at the Hort Farm. And of course, what's critical here is that you can see the soil temps rising and then falling over the time. And I've gone ahead and highlighted the 55 degree to 65 degree mark on this timeline, because this would be our targets. These would be our appropriate targets as far as when we want to go ahead and make these applications. So if we drag that down to the calendar, what you're looking at in the spring here is, let's say, first or second week of April through, let's say, the end of May as that spring window. And again, remember what's going on. The bent grass is waking up based on these same soil temps and increasing day length. We're getting to 11 and a half hours, 12 hours, 13 hours of day length in this window. So the bent grass is waking up. When its photosynthetic engine gets started, it sends down sugars to the roots. It wants to grow roots so it can capture water. Well, it also goes ahead and leaks some of that sugar out. And that sugar being leaked out is defensive. I mean, it wants to attract friendly things like trichoderma and pseudomonads and mycorrhizae, which are friendlies, which are going to give it some armor. But the take-all patch also recognizes these sugars. And again, the critical point about the activity of this organism and the timing of these preventative applications. And then, of course, we see in the summer we're outside of the normal windows. Yes, we know the take-all patch can still colonize and still be active all the way up to, say, about 80 degrees. But we know that that rate of spread is very drastically slowed down. So again, your preventative window spring is going to be probably in here, in this example at Rutgers. And if we look in the fall, we can see the same thing. So here we are right now, this week, you can see that the soil temps are just hitting 65 based on this data from last year. And of course, the temperatures go all the way down below the lower line at 55 by the beginning of November. So using this data, we now have some very clear application windows that we can go through. If I expand this to other data sources, so you guys can pull this up. This happens to be the National Weather Service, the Mid-Central River. And these are soil temps. I pulled this off this morning. These are actual soil temps in these areas that you can see in the dots. And you can see ranges anywhere from mid-60s, upper 50s in that area, which tells you what? It's time to make applications for take-all patch, is what it tells you. So we have these critical pieces of information which help us in the timing of our applications, and we know these based on the biology that we've been able to study over time. So when we think about curative and preventative activities, we have to think about integrated pest management. We have to think about the cultural and the chemical management practices and how these work together. So common things that we know about cultural practices is that take-all patch will grow better at pHs above 6.5 and will grow slower below pHs of 6.3. So of course, understanding that, and then we can think about, well, wow, isn't the same thing we found out for summer patch? And the answer is yes, it's the same thing we found out for summer patch. But this organism does have a pH range, which if we can achieve, we can slow down the activity of this organism just by controlling pH. And of course, we can control pH by the use of lime. We can control it by the use of our nitrogen fertilizing sources, thinking more about ammonium-based, ammonium sulfate, which are typically acidifying types of nutrient adds for nitrogen. One of the other key things, and this is where we differ a little bit from Summer Patch, is that it appears, based on our agricultural experiences, that Take All Patch can interact with the available manganese in the soil. And what it'll do is render that in an insoluble or unavailable form to the plant. And what that will do will cause localized or short-term manganese deficiencies for the plant. And of course, that benefits the Take All Patch organism, but can damage or reduce the functionality of those roots again for bentgrass. So what we have found over research is that the application of 2 to 3 pounds of manganese sulfate, not mag sulfate like we would use for Summer Patch, but manganese sulfate can help us go ahead and suppress this disease. And of course, we need to think about our soil testing. If we have a history of Take All Patch in the area, we probably want to be a little more diligent to those manganese levels in the soil to make sure that we're not in a deficient state. And if we are in deficiency and are seeing symptoms, then there is a cause and effect there. And some of that can be rectified by applying additional manganese sulfate at the rate of 2 to 3 pounds per acre in those areas. One of the things that we know about that would work best would be the spring applications of that, because that's going to be the onslaught, if you would. And of course, we want to make sure that we're watering that manganese sulfate into the active root zone for 2 reasons. One, it needs to be located into the root zone to affect the organism, which is a root zone organism. And 2, we want to get it off the leaves to reduce any foliar burn potential. Another thing, try to avoid, if we can, the excessive nitrogen. We've all been versed on this, and most of you are very well attuned to the spoon feeding situations that you go to. And I really want to highlight that in the next slide. As far as when we have a compromised plant, we need to manage it differently. Another thing to think about is that, remember I stated that this organism will overwinter as vegetative mycelium on plants. So we go ahead and we do things like aerating or verticutting. Of course, we have now have the opportunity to pick up materials, leftover mycelium, and move them. So my suggestion here is to think about this just in a physicality. Maybe you want to aerate or verticut your known areas of Take All Patch activity last. In other words, don't go through the Take All Patch and then go to an area that does not have Take All Patch, because you have the opportunity of physically moving pieces of organic matter, roots, thatch, etc., which may have mycelium on it, and then you're re-inoculating a new area. So it's one of those little things that you can do if you know where your Take All Patch is, and most of you do. Where it's located. That this is one of those little things you don't want to physically spread it. And then, of course, we get into the cultural practices of a dysfunctional root. And of course, all of you, if it's early June, you have to start thinking of August mode, because you have a short root system, you have a compromised root system, and one of the biggest things it's struggling for first is water to keep itself cool, and then secondly for nutrition. So doing those things that you would think about in August to keep your grass alive at all costs are going to be the same things that you'll try to do, let's say in June, when this disease first shows its head. Is try to think about your watering practices to keep the available roots that are still alive, to keep them wet. Think about practices that you can do for your stand health, as far as drainage, irrigation, we talked about pH, we talked about thatch and aeration, but all of these things, in other words, practiced agronomy to grow the roots. When we start to think about the chemical management of this, of course, we have to think about the products that we're going to choose. And of course, one of the first things that should pop into your mind is we talked about the timing aspects of these chemistries, but then of course, we have to think about the location of these chemistries. In other words, a root organism needs to have the products placed into the root zone. And if we look at the historical data, we see that most of that has been done with the DMI type fungicides, with the QOI type fungicides, and with things like FRAC1 thiophanate methyl. And of course, there's some very interesting reasons for that that we can talk about. And one of the ones that I would really point out to you very quickly is that, again, depending on the product that you have solubility and soil longevity characteristics that are going to differ by active ingredient. So propiconazole, metconazole, tebuconazole, prothioconazole, they're all going to vary slightly in their solubility, their ability to be drawn up by roots, and their ability to be washed below the root zone based on solubility in a sandy environment or a low organic matter environment, as well as what is their natural soil half-life based on other environmental factors like decay, hydrolysis, etc. And that really leads us into understanding that the DMIs are not all the same and the QOIs are not all the same in those principles. So what you're going to see when we talk about recommendations is making sure that if we use these products, that our application intervals don't outstretch the typical soil half-lives, and that the location of these products is in the right area for the root zone at the time. You see thiophanate methyl up here. And thiophanate methyl is one of those curious things because its activity in the plant, remember, is to reduce the cell division of the target fungi, of the target pathogen. So in this case, when we have thiophanate methyl exposed to the take all patch, the Gammanomyces, it tells that fungi, don't make more cells. So I want you to think about that in a sense of when is that chemistry going to be placed best? And my advice to you is that if you're using a thiophanate methyl type product in the management, a 3336 or other material, you want to make sure that you're using that in your fall programs, where you're trying to prevent infection into the new roots which are being formed as that bentgrass plant is doing what? Storing carbohydrates for the next year, making a sugar-rich root full of carbohydrates. So we're trying to prevent in the fall, and thiophanate methyl's best place is there, not in the spring. The DMI and the QOA fungicides, again, depending on which ones you choose, can be used in both windows, and that's one of the key parts. The other thing to remember is to make sure that we get these things located to the root zone and to time them based on what we know of your local soil conditions, to time them to the peak activities for prevention of colonization. So most of the university research is all generated down to some very key points, that application timing, which we really touched on, the reapplication intervals, again, based on the chemistries you've chosen, and of course, what we know about just about all of our QOI and DMI products that have been tested is that they will fit well into a 21 to 28 day soil reapplication window. So if we think about our soil timings and we think about a 1 or a 2 app program, and in most cases, we like to think about repeat applications depending upon the severity and how much we've got going on, that you want to keep those within that 21 to 28 day window during your time intervals. The other thing that research has pointed out, and of course we've seen this over time with various chemistries between protectant materials, whether they be local penetrant materials, whether they be upward moving systemic materials, is what water volume does it take to locate them to the right space? And of course, we think about take-all patch as a root infector. The first question is, what is my root zone? And of course, if I go out and take a profiler on 5 different courses, I may find 5 different root zones at the same time of year. So you need to measure that at your own course and look at what your root zone is, and then think about the water volume that might be needed to make sure that the chemistry gets moved into that active root zone. We know that high volume sprays historically have been done to make sure that we get that into the plant. I'm well aware that common practice of using high volume sprays is very limited on the application equipment and time and labor that you have. So most of you are in a regime where you will make the application and come back and irrigate that application off of the leaves, making sure that it gets through the crown and into the root zone. And of course, the data that we have will roughly tell you that between the 1/8 of an inch and 1/4 inch of irrigation is something that you should consider based on your conditions. And of course, that's all going to change individually for your green construction, your perk rates, etc., your organic matter content. So you'll have to adjust that. But in general, as a guideline, we think of that 1/4 to 1/8 of an inch after the application. When should I water? Always a fun question. Well, remember that some of these materials, the QOIs and the DMIs and thiophanate methyl, for example, all have the ability to foliar absorb. So we don't want to wait a long time. Yes, they do vary in how much foliar absorption there is, but we don't want to delay a spray in the morning and wait 2 days and then water it in. Something like that would not be efficient because we're going to lose some of the ability to get that material into the root zone because these are only upward moving materials, systemic materials. They're not downward moving. So we have to make sure that the watering practice that fits into your schedule, but it should be done relatively soon. We always get this fun question of people saying, well, I had take-all last year and I am I going to start a program, and how do I tell if a program is actually working? Because all of you have probably experienced, if you're on this call thinking of take-all patch, you're probably thinking, well, I had take-all, I haven't seen it for 5 years, and then this summer I saw take-all again. And why the interaction? Well, again, remember what we talked about. There's a lot of competition in the soil profile, the biotic profile against take-all patch. We know that take-all patch is competing for those root exudate sugars with a lot of other organisms. We know that some alliances and some antagonisms form. And of course, when the soil root zone, the biotic area changes due to chemical treatment, heavy sand top dressing, pH changes, all sorts of things that can affect that balance of biology can change. And of course, that's when take-all patch, being native, can take advantage of that and become active again. One of the things, of course, we always talk about in programs is that if you're going to do a program, how do I know it's working? How do I know what happened? Well, my simple answer is make sure, like we do in university trials, you leave an untreated area. I mean, it is a sacrifice to leave an area, but as best you can, leave an untreated area so that you can compare what did I do and what's the cause and effect of what I did. Very simply. So we start peeling off into data sets. And of course, I just pulled a few pieces of data. I didn't want to bomb everybody with data, but you give you some examples of some abilities of materials that are here. So this is a study from Steve McDonald. It goes back to 2006. There's actually older studies with Ned Tesseract and several other leading pathologists at the date. But what you're looking at here are, and I'm giving you some data to compare the preventative aspects. In other words, if I make applications late preventative, early curative in the spring, are there any advantages to fall applications, spring applications, product choices? So what you're looking at here with Steve McDonald's work are applications made, and you can see that he did 3 applications. You can see that they're all about 14 days apart. And he used tebuconazole. He used tartan, which is a mixture product of triticonazole. He used azoxystrobin, he used piracostrobin, and then he used headway, which is a combination of azoxy and propiconazole. And what you're looking at here is that these fall or these spring early preventative, late preventative, curative apps, again, Steve McDonald would be close to that data that we saw from Rutgers as far as that timing window. And I would tell you that what you're looking at here is an overall curative effect. 2 things to notice. Notice, one, that the untreated is declining over time. How come? Well, remember that the grass is going into summer, so the root sugars and the optimum activity timing or temperatures for this organism are declining, so it's becoming less active. So that's number 1. But number 2, you can clearly see that the choice of DMI or strobilurin in chemistry did have an effect. As you can see here in July, you can finally see that it's catching up and having an effect, on suppressing this disease. But again, this would be an example of curative at best. If I move that over to moving the application slightly earlier, this is some Steve McDonald work that he repeated in 2010. And what you're seeing here is the disease going through its ramping up stage during this year's soil conditions, and it's quite active activity. But again, you're looking at the DMI fungicides in particular here across this board, suppressing this disease to quite a manageable level. So again, you would call this the spring application, probably late preventative, based on the Rutgers timings and where Steve does his work. But again, you're seeing a cause and effect here with the DMI fungicides, giving you some very good suppression, over 60%, 70% suppression of this disease. If I move this again to think about other places, timing these applications slightly different. Again, oh, did it move? No, it didn't move. Sorry. Here we go. So I look at this and say, all right, so what if we do different applications, at different years? This is 2011, and we start to think of combination materials and what's going on. Here are applications initiated in April 26. These were 2 treatments at 30 days apart, 32 days apart. And again, you're seeing the strobilurin materials and the DMI materials. You've got Pinpoint, which is mandestrobin, Heritage is oxystrobin, metconazole, and a mixture of metconazole and mandestrobin. And you're seeing how we're able to really knock this disease down with these applications. Again, slightly earlier, but better in that window based on the Rutgers soil temps that I showed you, much better in that window of preventative in the spring. Looking at that study in a different way, again, you're looking at a second year. This is 2012 versus 2011. And again, you're seeing the DMI type materials and the strobilurin materials really suppressing this disease, giving you that knockdown. So we've got tools, and you'll notice that these are some of the same tools that we would use for summer patch. And you'll notice that these are some of the same soil temperature timings that we would use for summer patch. So it becomes quite interesting understanding that these are related in the same cousins, if you would, in the taxonomy of these fungi, but you're seeing these similarities, and we're seeing that properly timed applications with appropriate materials can give us some very good management. So as I wind this down, I hope I've given you, a good understanding of how this pathogen works, what its timing windows are, the fact that it's natural, it's there, it's competing in the biosphere with other things. And at times when it can outcompete the other antagonistic organisms, it moves from a saprophyte over to a parasite, increases its damage level, which then, of course, root dysfunction and plant death. We talked about the cultural practices as far as how they're going to integrate with the chemistries, the pH, the manganese, thinking of the organic matter removal, which is an inoculum remover, thinking about maybe doing the known patches last and doing all the other areas so that we don't take a, quote, dirty piece of equipment into a clean area, things like that. I hope I've shared you some of the turf research. Again, I kept this very short. There's probably 20 or 25 slides that if you go and do a web search on, you can find and get that material. And then, of course, really, I wanted to drive home the integration of these systems, knowing when it's active, knowing that we're doing preventative to stop root inoculation and root dysfunction, thinking of the fall timing window, thinking of the spring timing window, thinking of the active ingredients that we have the most history with. For technical support and materials, we have a technical piece in our flavor, if you would, talking about take all patch and capturing some of these many salient points. We do have our whole golf brochure. If you request, here's a series of references that I used in developing this talk today, as well as my historical educational background that you can all find if you do a quick search on the web and use these materials for your own education. And of course, if you need to get hold of myself or our tech team, we are available to you. There's three of us, myself, Aaron Hathaway, and Jason Fossey, which are all here to help you and support you, as well as our whole field staff of our local representatives, which if you have a question, you can reach out through your distributor or directly to Nufarm and ask the technical questions so that we can bring answers to bear. And with that, I'll see if there's any questions that we can take or any other issues. So Ruel, over to you if there's any questions that I can't see in the chat.
All right. Thank you, Rick.